Welcome to Lesson 14.2, Human Genetic Disorders. In this lesson, we'll explain how small changes in DNA cause genetic disorders and summarize the problems caused by non-disjunction. Changes in genes' DNA sequence can change proteins by altering the amino acid sequences which may directly affect a phenotype. There is a molecular basis for genetic disorders. Thousands of genetic disorders are caused by changes in individual genes. These changes often affect specific proteins associated with important cellular functions. One disorder caused by individual genes is sickle cell disease. This disorder is caused by a defective allele for beta globin, one of two polypeptides in hemoglobin, the oxygen-carrying protein in red blood cells. The defective polypeptide makes hemoglobin a bit less soluble, causing hemoglobin molecules to stick together when the blood's oxygen level decreases. The molecules clump into long fibers, forcing cells into distinctive sickle shape. Sickle-shaped cells are more rigid than normal red blood cells, and they tend to get stuck in the capillaries, the narrowest blood vessels in the body. If the blood stops moving through the capillaries, damage to cells, tissues, and even organs can result. Another disorder caused by individual genes is cystic fibrosis. The most common allele that causes cystic fibrosis is missing 3 DNA bases. As a result, the amino acid phenylalanine is missing from the CFTR protein. The normal CFTR is a chloride ion channel in a cell membrane. Because of the missing phenylalanine, the CFTR is abnormal and cannot transport ions across the cell membrane. Consequently, the cells in the person's airways are unable to transport chloride ions. As a result, the airways become clogged with a thick mucus. People with one normal copy of the CF allele are unaffected by CF because they can produce enough CFTR to allow their cells to work properly. So, 2 copies of the defective allele are needed to produce the disorder. What does this mean about the CF allele? It means that the allele is recessive. So why isn't the cause of CF considered a frameshift mutation? Well, because exactly 1 codon, 3 nucleotide bases, have been deleted, so the rest of the reading frame has not been affected. Huntington's disease is another disease caused by an individual gene. In this case, it is caused by a dominant allele for a protein found in brain cells. The allele for this disease contains a long string of bases in which the codon CAG, coding for the amino acid glutamine, repeats over and over more than 40 times. So why are these alleles still around if they can be fatal for those who carry them? Well, they provide some evolutionary advantages or benefits. Most African Americans today are descended from populations that originally lived in West Central Africa, where malaria is common. Malaria is a mosquito-borne infection caused by a parasite that lives inside red blood cells. Individuals with just 1 copy of the sickle cell allele are generally healthy and are also highly resistant to the parasite. The resistance gives them a great advantage against malaria, which even today claims more than 1 million lives every year. About 100,000 people worldwide die annually of sickle cell disease, but malaria kills over 1.5 million. So from a population perspective, the benefit of having the allele outweighs the drawback. More than 1,000 years ago, the cities of medieval Europe were ravaged by epidemics of typhoid fever. Typhoid is caused by a bacterium that enters the body through the cells in the digestive system. The protein produced by the CF allele helps block the entry of this bacterium. Individuals heterozygous for CF would have had an advantage when living in cities with poor sanitation and polluted water. Because they also carried a normal allele, these individuals would not have suffered from cystic fibrosis. Let's review meiosis. Most of the time, the process of meiosis works perfectly, and each human gamete gets exactly 23 chromosomes. On occasion, something goes wrong during meiosis. The most common error in meiosis occurs when homologous chromosomes fail to separate. This mistake is known as non-disjunction, which means not coming apart. If non-disjunction occurs during meiosis, gametes with an abnormal number of chromosomes may result, leading to a disorder of chromosome numbers. If two copies of an autosomal chromosome fail to separate during meiosis, an individual may be born with 3 copies of that chromosome. This condition is known as trisomy, meaning 3 bodies. The most common form of trisomy involving 3 copies of chromosome 21 is Down syndrome, which is often characterized by mild to severe mental retardation and a high frequency of certain birth defects. How could Down syndrome result from the type of non-disjunction shown in the figure? Down syndrome could result if one of the 2 cells on the left met up with a normal sex cell and the resulting individual had trisomy for chromosome 21. A female with Turner's syndrome usually inherits only one X chromosome. Women with Turner syndrome are sterile, which means they are unable to reproduce. Their sex organs do not develop properly at puberty. In males, non-disjunction may cause Klinefelter syndrome, resulting from the inheritance of an extra X chromosome, which interferes with meiosis and usually prevents these individuals from reproducing. There have been no reported instances of babies being born without an X chromosome. What does this fact indicate about the X chromosome? It indicates that this chromosome contains genes that are vital for the survival and development of the embryo. So in summary, small changes in a gene's DNA sequence can change proteins and may affect our phenotype. Non-disjunction may cause gametes with an abnormal number of chromosomes.